Good morning, everybody. Can you hear me okay? I'm my name is Sandy McEntee. I'm logged on as Amber today to lead this meeting because she could not make it. Um, I am an SLC 6A1 mom to my daughter Katrina. Katrina is 17 years old and she was diagnosed when she was 13. I have been helping Amber for the past 3 years, just about 3 years now, helping her, recruit for the patient registries and with the website and other various projects. This is my first time hosting this meeting, so be patient with me. Let's see, let's get this going here. Um, can everyone see the slide here?
Yeah, we can see it.
Okay. Of course, Amber threw in this great dad joke. What type of dogs do chemists own? Uh, Laboratory retrievers. Um, our mission, SLC 6A1, the mission is, um, all right, I'm going to start over because this is I'm a little nervous, but we are a patient organization dedicated to improving the lives of children and families affected by SLC 6A1. Our focus is to raise awareness and fundraise to advance scientific research that will ultimately result in a cure. Today, I want to introduce Tara Bayat. Did I get your name correct?
Yes, perfect.
Okay. She is a PhD candidate at Texas Tech University. Go ahead, Tara.
Well, thank you so much. Good morning, everyone. It's my first time in this meeting, and it's a pleasure to be here. My name is Tara Bayat. I'm in the final year of my PhD in biological sciences in Dr. Clementina Fantasser's lab at Texas Tech University. My research is focused on the role of melanoma antigen L2 or MAGE-L2 in Prader-Willi and Schaaf-Yang syndromes. And I've been studying reprogramming of mRNA translation, particularly in the hypothalamus and pituitary gland using polysome profiling. I've also done single nuclei RNA-seq on the hypothalamus of MAGE-L2 knockout mice. And I was also interested in studying hormonal imbalance and stress response through HPA axis in MAGE-L2 knockout mice. And recently, I've developed patient-derived organoids in our lab. Well, I would love to continue my research on neurodevelopmental disorders in my postdoc, and would love to connect with you if you know of any opportunity. And thank you so much.
Thank you. Thank you, Tara. I wanted to let everybody know that the SLC 6A1 Scientific Symposium is going to be happening in December, I believe on the 4th and 5th in Atlanta, Georgia. We are currently nailing down the plans for the location. And it's time for everyone to register. I'm going to put the link in the chat here for registering. We are also, I wanted to remind everyone that we have travel scholarships available through Uplifting Athletes, and I'm going to put a link to that in the chat as well. Now I would like to introduce Lainey Mosley. She has worked on this amazing film and is going to tell you more about it. Let's see.
Hi. I will do a quick introduction and then show you an 8-minute trailer for the film. But I started this documentary 4 years ago during the pandemic, really when we were all, struggling to do something. And it came to me that I'm the mom of a daughter with a rare disease who has something called CHOP syndrome, and she's the oldest in the world with it, and she wasn't diagnosed until she was 18. She's now 28, and I was struggling to sort of look at, well, am I doing enough? What else? What are other people doing? And I ended up going to a boot camp at Ultragenics and realized that there was this whole new generation of advocates and kind of warriors fighting for accelerated science and research. And that was such news to me. I didn't know that existed. And so I decided that I thought it was really an important point in time that it needed to be acknowledged and celebrated, and also understood, and the rest of the world needed to be made aware about the importance of rare disease research. So that's the film. You'll get a little taste of it here. Hold on. All right, here we go. And I'll try and make it bigger.
From a rare disease called HNR-NPH2.
Oh, wait, let me just start over.
First, from a rare disease called HNR-NPH2.
I think, Lainey, we can't see your.
Doesn't
your
talk.
screen.
She struggles with walking.
Wait, you can't see it?
No,
No.
it doesn't seem like you've shared your screen yet.
Oh, hold on. Oh, so I had to go back and share. Okay, hold on. All right. All right, you can see my screen now. Can you see the screen?
Yes. Yes.
Okay. All right, sorry about that.
And then just enlarge.
Rosie is my youngest daughter, and she suffers from a rare disease called HNR-NPH2. She doesn't talk. She struggles with walking. She may go catatonic when she reaches puberty. The clock is very much ticking for us.
There's huge unmet clinical need. There are lots and lots of rare disorders for which there is absolutely no treatment.
This is recorded by Brandon Rojas. Here we go. Yeah, I can stop a little bit and start going.
Brandon was diagnosed with a rare disease called adrenoleukodystrophy that had affected since birth, but we didn't know until he was 7 years old.
I am a dad to a little girl called Hassie, who's 8 years old. She suffers from a rare genetic condition that is life-limiting and life-shortening. I love you, Hassie. The geneticist told us what CDLS meant, and I said, okay, how do we fix this? And she said, there's nothing you can do. This is genetic, and that's...
Oh my God, Mom.
Oh my
Look
God.
how pretty you are. Okay, so this is my mom. She didn't always look this way, but she's 50 years old and she's hotter than most.
Wow. Okay.
Maxwell's missing one half of a gene that controls the body's most important inhibitory neurotransmitter. And they said no medical doctor is going to be able to help you. You need to find scientists. We're in a fight against time. He must be treated in the next year before he'll suffer irreversible harm to his neurological system.
The vast majority of rare diseases have a completely uncoordinated, disorganized, sort of like, let's hope that the stars align approach to research.
Most of the funding for biomedical research comes from the National Institutes of Health, but they tend to focus on diseases that affect a lot of people, things like cancer, cardiovascular disease, things like that.
If you have less than, say, 1,000 patients, companies are kind of like, there's other diseases that are going to make us a lot more money.
So the only people that actually take on that work are patients. Come on in, Carrie.
So excited.
This is where it all happens.
Lily has half of what she needs in her gene TSHZ3. And we believe that this insufficiency in her gene is what's causing her autism.
It's on the microscope.
There you go.
You have to learn genetics, you have to learn drug development, you have to learn all these things that people literally got PhDs and MDs for.
It requires so much alignment and this realization that we can actually do something.
My name is Amber Freed, and I am the founder of SLC 6A1 Connect. I'm also fighting like a mother to give my son a chance at life. It's been a pretty wild ride. I've raised close to $5 million philanthropically. We've repurposed a drug. I had a mouse genetically engineered of my son. I'm not going to stop until my miracle happens.
Because we are getting to the point where we can see it spelled out in the DNA, sometimes it's a single letter that all we would need to do is figure out a way of targeting or fixing or being able to compensate for that one letter. There are incredible scientists who are working right now to be able to come up with ways of compensating or correcting for that.
So these are rosy cells.
A lot of times we think of genetic mutations as loss of function. We know there's a mutation, we know that it destroys the gene, and sometimes the easiest thing to think about is, why don't you just put it back?
Rosie has one good, what we call an H2 gene, and one bad H2 gene. So we're just going to try to take the bad one out. It'll cost us between $500,000 and $1 million.
Toxicology is one thing. Also, manufacturing the vector seems to be the biggest cost.
How much did you say?
3 to 5 million.
Sort of money that is just staggering for a normal family. We are at 390 something grand.
We have to fight to get closer to a cure for my daughter and these other kids.
You are warriors, and this is a battle. You will need endurance. You'll need strength. And no matter what happens, you are heroes for taking on the battle.
Slow it down when you ask for the money, or slow it down when you say critical things. This disease is pause. This is critical.
Rare diseases are not all that rare. They account, if I were to go across the street in the hospital, to about 50% of the children we have in the hospital right now.
In medical school, it's never really like summed up to be like, well, actually, there's 30 million people with a rare disease. There's 10,000 rare diseases.
And I want to tell you a little bit of story why we're here and why tonight is so absolutely important.
I tend to try to gravitate away from using the word rare disease because that instantly just turns people off. The normal person hearing it's just going to be like, why should I care about this?
By changing the life of one little girl, we can change the lives of millions.
We turn now to Ms. Patricia Furlong, president of the Parent Project Muscular Dystrophy. Regrettably, both of her sons died of muscular dystrophy.
If the universe is going to take my children and not let me see them grow up, I'm going to get even with you and I'm going to try to move the needle to the degree that I can. Our children are not out of their warranty period before their bodies wear out. They will never receive adult status to advocate on their own behalf. Nothing will change without increased investment in Duchenne research. We talked about legislation. We talked about making friends in Congress. We talked about sort of a stepwise process. So we found some friends. We wrote on the back of an envelope the wish for a piece of legislation that was introduced into the House and Senate and signed into law in 2001. We seek equity in research opportunities, equity in set-aside funding, equity in the review process, equity in worth, the worth of every child with Duchenne and Becker muscular dystrophy. Mr. Chairman, it's too late for my own sons, but with your help, this disease will change.
It's never a good time to be diagnosed with Duchenne's. But if there's ever a time, now is the time to be diagnosed.
Today, the committee will meet in open session to discuss the biological.
First gene therapy in Duchenne muscular dystrophy.
Do you think that's going to be a challenge to demonstrate to the FDA?
We're still thinking about rare diseases like common diseases, clinical trials with placebo controls and dose escalation. And when you have just a very small patient population, you can't think like that anymore.
Waiting is the worst thing that we can do because we don't have time.
As you can see, even a 1-point change in the North Star is clinically meaningful.
My two sons both have Duchenne muscular dystrophy.
While I'm speaking, you will see comparison videos of Bracken before and after receiving gene therapy.
Please approve SRP 9001.
More certainty of Duchenne untreated. Danny, gone at 16. Austin, gone at 16. And Mitchell, gone at 10.
It's a terrifying start, I must say.
The scientific advances are there. The technologies in CRISPR-Cas9, that's there. And you can show, a mountain full of improvements. But when it comes to getting rare diseases into the clinic, we haven't made very much progress at all.
Children shouldn't be sick, right? They just shouldn't be.
Did you do
Yeah, but why do we have to do all this?
We don't have much, but whatever we have, we're putting it in. And a big part of that is us. Our mental and physical ability is all on the table.
We're looking at like 150 million kids, I guess.
Hi, you know mommy loves you? Yes? How much? Here to the moon. Yes? You love mommy too? Yes?
I want to participate in these children not being overlooked anymore, not being forgotten.
Thank you, Lainey. That was amazing.
I never post stuff like this, but I had to share.
Stop. Sorry. All right. Let
Do
me
you mind,
stop sharing.
do you mind if I make a comment as a person who's worked on rare diseases for 20 years?
Yeah, sure. Go ahead.
I've worked on duplication 15q, Angelman, Prader-Willi, Rohad, CCHS. You know, rare disease is my wheelhouse. Okay. And I really, really respect the families and met many families with many of these syndromes. And that fight that you guys are fighting now isn't going to get anywhere if you don't fight the government right now. 40% cut in NIH funding and cut of the indirect costs to the institutions and the war on science is going to make all of this effort move backward.
Yep.
So the fight needs to turn from rare disease to science. I mean, if you kill science in this country, no one's going to be working on anything. And, you know, the big black cloud is on the horizon.
Yep. We're all terrified, but yeah.
Not as terrified as us and everybody in my lab, I think. We're directly terrified. You know, people are losing their jobs. 180 people got canned at Columbia the other day. And I'm not going to sit back and watch it, you know. And I think you guys are fighters. So maybe you guys are the ones who should fight, right? Because, you know, you're not scientists. You come from the perspective of people who understand why we do this, why we do science, why we work on rare disease. So somebody needs to tell people in charge because things are falling apart.
Thank you.
Thank you. All right. Thank you, Lainey. That was amazing. I highly encourage, you know, would just get the word out about this film. It's incredibly moving and really highlights the level of effort that we're all putting in, all of us. Okay, I'm almost done with the announcements here. I did want to mention this. The Biotech Club at UT Southwestern is hosting a panel on SLC 6A1 called Rare Disease, Real Impact. It's happening the evening of May 30th. The panel will be made up of SLC 6A1 advocates and Steve Gray. Everyone is welcome. If you're in the Dallas-Fort Worth area, please join us. There are 3 new papers that have been published. There is one, I'm not going to read these. Just take a look, and then I will put the link to each of these papers in the chat. Let's see here. Our next meeting in June we're going to be having, I hope I pronounce this correctly, Kaila from the University of Helsinki. He is a self-proclaimed scientist who is restless and built a career on electrophysiology of GABA. I would like to ask if we have anyone new at the meeting today. You can introduce yourself like right now.
Hi, I'm Lindsay Rosardi. I'm assistant professor at University of Alabama at Birmingham. I've been working with Cameron Crowder on an SLC 6A1 project. My lab focuses on generally functional genomics of neurodegenerative diseases, but we're experts in iPSC models. And so with Cammie's pulled us into the SLC 6A1 world. And so we're modeling some patient lines now, trying to really understand directly what the GAT transporter is doing and how GABA inhibition can modulate that.
Welcome. It's nice to meet you, Lindsay. I'm glad you could be here.
It's great to join. Thank you.
Is there anyone else that's new today?
Hello, actually, I'm Anil. I am from the Jackson Laboratories Rare Disease Translation Center. I am joining Jensen Miguel's group, and I'll be working with her with the SL6A1 projects as well. So it looks like a very nice group, and I'm glad to be here. So hopefully we'll be working together as well.
Awesome.
Thank you.
Awesome. Welcome, Anil. Great.
I am also new here. I'm actually the aforementioned Kaila.
Hi.
And right, yeah, and yeah. So I have a long career in GABA transmission, and this sort of I would say actually the SLC6A1 mutations, they come like directly on into the like main area of research which I have done, because I mean we are looking actually like we will look at the acetylcholine effects, which are really like they are proven to be really efficient, this effect in patients. So we will dig out the mode of action of acetylcholine and also try to maybe improve medication, because there's a long way. It's really a long way until this sort of gene technology is being developed. So we need really good drugs before that. And this is a really promising field, like carbonic anhydrase inhibition. So yeah, so I'm not sure what proclaimed. What did you say? I don't know. How did you actually say? What did you actually say about me, my career? Like you had some sort of adjective there.
It might have been Amber's notes. Just that you built a career on electrophysiology.
Yeah, sure. Okay, so I have done molecular biology, electrophysiology. We have lots of papers on birth asphyxia, on neonatal EEG, also like adult seizures. Quite a lot of like research on all levels of biological organization, also behavior. So like I said, I mean, like it will be. I mean, you're getting these mice very soon.
Wonderful.
So I just really look forward to starting work to work on them. Within 1 month we don't get any data. So my talk in June will be more like about the background mechanisms and so on. So
Okay.
and yeah, well, I'm not. Some people actually in the audience know my name at least. So. But the scientific career has been long. I have been 3 times academic professor in Finland. I have got an ERC grant, etc., etc. So there's quite a lot of experience locally. So happy to join this crowd.
Welcome, and we
Thank
look
you.
forward to like hearing more about you and your work next month.
And I look a little bit older than you had in the photo.
This is my second meeting. I actually came to one of these before, but I guess I should introduce myself. And I actually know Lindsay because I saw her talk here at Tulane recently, and we talked quite a bit. I really like what she's doing. And that actually is part of my involvement here. So two things that I do. One is I use Drosophila to screen for anti-epileptic drugs. And we've done that successfully with duplication 15q syndrome. And we're moving into doing other fly homologs that we found for human epilepsies. And we're putting those in our pipeline and trying new approaches to natural products and other FDA approved drugs. So repurposing drugs instead of going in the same direction as common anti-epileptics that people use. And then the other part of my lab makes dental pulp stem cells from rare disorders. We have over 200 dental pulp stem cell lines from 15 syndromes. And we were in the process of trying to get permission from my IRB here to collect SLC 6A1 teeth and make dental pulp stem cells, which we can make into neurons. And we've done lots of things with these neurons, circadian rhythm studies, molecular studies, so I'm excited to be involved with this group, and I would like to continue to work in this area.
Thank you. Nice to meet you, Larry. I'm glad you joined us again. Is there anybody else that's new today? Great. Okay.
Yes. Sorry.
Oh, I'm sorry. Go ahead.
So I'm Sabah Farhad. I'm working as research coordinator in medical college here in Karachi in Pakistan. And I have started working as a PhD student, and I have worked on subclinical hypothyroidism, the maternal one, and the effects, the neuropsychiatric effects on the offspring. So my findings were very interesting, and it helped me to go into the details of neuropsychiatric changes and neurodevelopment at the earlier stages. So therefore, basically, I got some interest in this SLC 6A1 project and your efforts. I really admire Amber, you basically, because you're a very strong woman, and I have found that yes, these rare diseases, which are not really rare, I have also have an experience like a family member of a person who are living with the rare diseases. One of my brother is having Eisenmenger syndrome, and the other is having epilepsy. So I really know the consequences and the challenges we all have on the daily basis. So a very nice effort, and I would like to continue to be a part of this group. Thank you so much.
Wonderful. Thank you. I'm glad you can make it, Saba. And I'm not Amber. I'm Sandy.
So
I'm just
the name on the screen is
I know that. I'm posing as Amber today. Yeah.
Okay.
Okay. Let's move on to the main attraction here. Franco Lombino is going to talk about cell type specific intracellular roles of Gat1. Franco, would you like
to thank you.
Over? Yeah.
I will try to share my yes, now my screen. I hope you all can see in a presentation mode.
Yes.
Okay. So thanks a lot for having me here speaking today. Actually, the title was a bit longer. It was maybe truncated, maybe because of space issues. But actually, I would like to talk about intracellular roles of Gat1 in modulating neuronal activity, but also about an unanticipated extracellular localization of this transporter, at least at the brain level. So my name is Franco Lombino. Yes, I'm in Novarino's lab at the Institute of Science and Technology, Austria. So we are close to Vienna. Not really in Vienna is the institute. This is how it looks like. I will try to cut the story a bit shorter due to, I don't know, the time availability that we have. But basically, our building is this one here hiding behind the tree. I will just, sorry, I think I need the pointer. It's a very nice environment to work in, yeah, and the institute is constantly growing. So just to briefly introduce myself, I started my PhD in 2015 in Germany after a brief experience in New York where we were studying Alzheimer's disease. During my PhD, we were focused primarily in basic science and applied to synaptic function, yeah. And then I joined Gaia Novarino's lab, where we are actually studying neurodevelopmental disorders from at least a synaptic perspective. Yeah. So I will talk today about extracellular vesicles, and this is how I connect this transporter to the extracellular space, are around 100 nanometer vesicles that are secreted by almost every cell type known. Yeah, and this is how they look like at the spin in this microscope live in solution when you actually label them with a lipophilic dye. In this case, it's dye labeled. You put it in solution, you look at the microscope, and if you're lucky, after X hours, you see a vesicle swimming around. Yeah. Yeah, but this project actually has a beginning with one of my former colleagues, which is Dr. Oliveira, who basically isolated these vesicles from the cortex of mice and performed the developmental profiling of the proteome of these vesicles. And actually, she observed that the protein content of these vesicles is actually tightly regulated during development, yeah, indicating that at least at the protein level, but she also focused on other molecules, yeah. These vesicles seem to have a function in the brain. Yeah, these vesicles have been extensively studied in other fields, such as the cancer field. Yeah, they were first observed quite some years ago, but actually there's a boom now in the last years where the interest on these vesicles has dramatically increased, due mainly to the fact that now we know that they have a function. Yeah. The origin of these vesicles, I don't know how many of you are familiar with them, is endocytic, yeah, primarily. Therefore, you get the endocytosis. You get endocytosis of the plasma membrane, the pinching in of the membrane. Then a multivesicular body forms that then fuses to the plasma membrane and releases to the extracellular space the vesicles that are encapsulated in this multivesicular body. Yeah. So now, this is a brief description, but now we know that, or the field considers these vesicles as a message in a bottle. Yeah. So, like in an additional way of communication between cells of the brain. Yeah, and this is just to give you an idea about how complex this system is, because we know in the brain we have different cell types. Different cell types secrete different vesicles that then can target different cell types themselves, and therefore mediate different effects in target cells. Of course, these mechanisms are now observed, but they are not still deeply characterized. So we still don't know how this occurs, and if the process is tightly regulated, if the migration of these vesicles in the extracellular space is targeted, or if it's a stochastic swimming around of these vesicles and targeting stochastic cells. Yeah. I have nothing to say about Gat1, of course, in this group, because we all know, but for me, or for this talk, it's important actually to remember that mutations in SLC6A1 primarily cause impairment of GABA uptake into the cell. Yeah, but ways in which this is produced can be different. One is because these mutations actually affect GABA binding, but also because, at least in some of these mutations, the trafficking of the protein to the plasma membrane is affected. Yeah. So now we identified I mean, we managed to isolate these vesicles, and this is how they look like at the electron microscope. Yeah, I mean, these images actually are quite astonishing also for the field. I would say we can actually immunolabel these vesicles with specific vesicular markers, CD9 in this case, that is an exosomal or extracellular vesicle marker. And this is actually how they look like at a higher magnification. And what is surprising for me is that one can actually almost see the lipid bilayer. Yeah, so also suggesting that these vesicles are intact after the preparation. Yeah, so that the content of these vesicles potentially is not lost during the preparation. So as I anticipated before, we isolated these vesicles from different at different ages of from the cortex of mice at different ages, and as you can see here in this panel, for example, at P0, yeah, we can see a massive increase of Gat1 in the F2 fraction of these vesicles. So this preparation is actually a sucrose gradient, essentially, yeah, very not super complicated. And the F2 fraction, so you basically, after the sucrose gradient, you isolate 3 fractions, and in the F2 fractions, you mainly have vesicles that are called exosomes that are of a diameter of around 100 nanometers, yeah? So there is a high enrichment of Gat1, but also an enrichment of CD81 that is a positive marker for these vesicles, and the lack of BIP1 that is a negative marker for these vesicles. So indicating that indeed our preparations are quite clean. Yeah. I will not spend time on the P30 time point because anyways, I mean, we have a switch to another fraction. We can hypothesize many things, like the cells that are secreting these vesicles is different, or that the vesicles themselves are different. In F3, we have even smaller vesicles. So, but our goal for the moment is to focus on these early vesicles. Yeah. We also generated human cerebral organoids. Yeah, so we started first with stem cells, and as you can see, we isolated the media. This is quite simple. Yeah, so you grow your organoid. I mean, not the organ themselves. To generate the organoids is quite complicated, but then the vesicular isolation is quite simple, in the sense that these vesicles are secreted in the culture media, and therefore you isolate this, you extract this media, you perform Weissen's, and you can see the presence of Gat1. When we actually subject this media to immune affinity purification with positive markers for exosomes that are CD63, CD9, and CD81, you can nicely see on this fraction the co-precipitation of Gat1 together with CD81 as well, yeah, so which is the positive marker for exosomes. We also obtained, actually from SLC6A1 Connect, as patient cells and CRISPR-corrected isogenic controls. We generated human cerebral organoids again, and basically we did the same experiment, okay? So actually, what you can see here is that there's no much secretion compared to the stem cells, actually, but when we immunopurify these media, we can actually see the presence of Gat1 too. Yeah, so potentially this is the wild type version of Gat1, also in the mutant. Yeah. So okay, now we know that in all our systems we can see Gat1 in the extracellular space. So we started using different models. We got in contact with Professor Trich, who was a frequent listener in this group. Actually, I don't know if he's here, who kindly shared this mouse with us, SLC6A1 flux animal, that actually would allow us to understand 2 things. First of all, cell type specific phenotypes that I think it's quite relevant for the field at the moment, at least to understand which one of the patient's phenotypes are actually associated with specific cell types. Yeah, but also to understand which cell type could be actually responsible for Gat1 secretion. Yeah, and why is this happening, at least at P0. So I also want to mention that being Gat1 in these extracellular vesicles could have an impact on the target cell in two different ways. So one possible way could be through the actual mechanism by which we know Gat1, that is the transport of GABA, at least. So we want to know if GABA is present in vesicles. But another possibility is that Gat1 itself is doing something in the membrane of the target cell by functioning specifically in the target cell, not necessarily in the vesicle. So the other model that we are using are just plasmids that we actually kindly obtained from Professor Sonja Zucic here in Vienna, who kindly shared these plasmids with us. And actually we became very, very interested, also aiming to understand how the secretion process happens. We became very interested on the R44Q variant and the A288V variant, yeah? So we know that the R44Q actually reaches the membrane but is unable to transport GABA, while the A288 variant is actually primarily trapped in the ER and also is unable to transport GABA, yeah? So we started testing all our models. First, we started with the plasmids. We used N2A cells. N2A cells are mouse neuroblastoma cells that actually we transfected with these three constructs, adding also the wild type version of Gat1. And we wanted to see if all of them were actually present in these vesicles. I mean, here you can see that we nicely recapitulated the data from Professor Zucic, where she actually couldn't see the mature form of Gat1, yeah, at least with this construct, the A288V, yeah. So, and then when you go to the extracellular vesicles preparation, you nicely see that this variant, the A288V, actually doesn't reach the membrane, is not packed, is not transported in extracellular vesicles. We cannot detect it. So for us, this was an interesting question or interesting aspect, yeah, because we could actually understand that potentially the presence of Gat1 at the plasma membrane is actually a prerequisite for this protein to be in these vesicles, yeah, which means also that potentially the healthy form of Gat1 is present in these vesicles. So actually to confirm this and also to make sure that we were actually observing a variant that was systematically trapped in the ER, we performed some live cell imaging. So here you can see, I will show you N2A cells transfected with wild type, with the wild type version of Gat1. Why I cannot play my video if I played it all the time before. Sorry, I need to understand how. Well, I can play it like this. You can still see it, right? Okay, so in live cell imaging, you can see transfer overexpressed Gat1 actually that moves quite dramatically, and doing time-lapse microscopy. And for those who actually did time-lapse microscopy, this is quite indicative of a microtubule movement, microtubule-based movement. Very fast, very directed, and everything is moving. So we decided actually to go for a turf microscopy. Yeah, so these are traces of chymographs of this movement. We decided to go for a turf microscopy. Turf microscopy, for those who don't know, is based on the tilting of the laser, the excitation laser, that actually manages to excite only some fluorophores close or in proximity to the plasma membrane, allowing us to observe only phenomena that happens within the deepness of around 100 to 200 nanometers in depth, yeah? So we moved to turf, in this case, and the same cell, and you can see now that the background is dramatically decreased, but also that the movement is more indicative of proteins or vesicles or patches of proteins that are in the membrane, yeah? So we also isolated here an example where you can actually see these vesicles that appears and this appears from the plasma membrane, suggesting that indeed it gets there, but it also gets internalized. Yeah. Yeah, and you can see a chymograph for this event. This, for those who are familiar, is indicative of an exocytosis, endocytosis mechanism, where there is a kiss of the membrane and then goes back. But then we also transfected or overexpress this variant, the A288V variant, that actually never reaches, or I mean, at least compared to the wild-type version, it doesn't reach the extracellular vesicles. Yeah, it's not secreted. So, and this is what we see. Yeah. So, and this is for those who did time-lapse imaging, this could be reminiscent of actually ER. Yeah, ER that is in close proximity with the plasma membrane of these cells. This is a static example, actually, where you can actually see these tubules that remind you of ER, and these are published examples of these two events. Yeah, so on the left, you can see vesicular turf, where you actually image the membrane, you see these patches, they move. And here, these authors actually expressed an ER pump, ATPase pump, where they actually managed to see the ER, and this is exactly what we see, yeah, indicating that actually A288V is present primarily in the ER. It doesn't reach the membrane and is not secreted via extracellular vesicles. We also went back to the cells that we obtained from SLC6A1 Connect patient cells, and we wanted to see if actually the mutant version, this mutant, S295L, was also enriched in the ER. So I think everybody here is familiar with the difficulties of a Gat1 stain. So, but I think we tried it, and the results are quite okay. We will repeat this and possibly improve it too. On the left, the first image is a wild type, when you can see a diffused staining, but also some vesicular patterns, while in the mutant cells, the other three cells, you can see some enrichment in the perinuclear fraction or in the perinuclear area. Indicative, of course, we need specific markers of ER accumulation. Yeah. And this is also for us a good indication in the fact that actually it's observed also by others. We are not actually discovering that these variants get trapped in the ER, but for us it's a good confirmation, and it's also a good indication that Gat1 needs to be in the membrane, needs to function to be released in extracellular vesicles. So we went a step further, and actually in the next 2 or 3 slides, I want to actually make sure I make this disclaimer. These are these are tests now that we are doing, and therefore that would set the basis now for the actual experiments. Yeah. So now we are having preliminary observations, but we really need to optimize and make sure that what we're seeing is solid. Yeah. So please take this with the forceps, and knowing that this is actually what we want to do. This is our direction, but it's not concluded yet. Yeah, and not conclusive. So we actually put in culture mouse primary cultures, yeah, from the cortex. And we let them sit for 12 days there in culture. And then we actually isolated extracellular vesicles from mouse brains, labeled them with dye. These vesicles that you saw swimming around are basically labeled in the same way. And we administered these vesicles to these cells. So for us, this is a proof that actually we can deliver these vesicles to the cells, that we can visualize them, and that they get actually internalized into the target cells. Yeah, one of the ideas, or what we want to do, is actually to observe if Gat1 levels, and this is super important for us. We need to dissect if these are endogenous Gat1 levels or vesicular Gat1 levels. Could actually be impacted by the delivery or the administration of these vesicles. Another thing that we can do is actually with these primary neurons is to isolate vesicles actually from the cortex of mice that are knocked out for GAT1. Yeah. So here you can see the SLC6A1 flux mouse that I talked to you about before, that actually, and now you don't see my pointer, of course, that actually is mated with the Cree driver line, the VGAT, to actually deplete GAT1 from all the interneuronal population. And here you can see in the first two lanes, the total lysates, yeah, and these are the F2 fraction that contains primarily exosomes, yeah. Here you have a control mouse where GAT1 is actually present like before, and then a dramatic decrease in the conditional knockout, indicative of the fact that interneurons are secreting GAT1, yeah, but that they are not the only cells that secrete GAT1 to the extracellular space, because we still see the band, yeah. Okay, and these EVs of these two populations, actually of these two genotypic backgrounds, can actually allow us to understand if GAT1, when delivered to the target cells via extracellular vesicles, actually play a role, not only extracellular vesicles themselves, yeah? So actually, this is what we plan to do to actually assess the GABAergic system function, like, for example, GAT6 as an enzyme, but also cell viability, cell stability, and also perform some electrophysiological experiments. Yeah, so primarily focusing on inhibitory currents. Yeah, so of course. Our goal or our understanding of these vesicles is that actually the vesicles themselves already play a role, and we really want to know if the depletion of GAT1 of these vesicles can also have an impact on the phenotype that we observe in the target cells. Yeah, allowing us to actually understand if GAT1 could actually modulate inhibitory neurotransmission in these cells. And this is the part that actually was in the title that was published or that was promoted. That is the fact that we also want to know what's happening or which cells are actually responsible for specific phenotypes in at least in the mouse. So we work with knockout mice also because we want to know protein function. Yeah, we are not trying to really mimic patients' situation. Yeah, so we know, you all know, this is just for us for confirmation. We know that GAT1 actually increases during development. And what we did is to actually use this flux mouse and cross it with different driver lines. Yeah. So first of all, in the upper part here, you can see the EMX3 driver mouse that actually depletes GAT1 from specifically pyramidal neurons, but also astrocytes and oligodendrocytes. Yeah. And this is something interesting because although we know that GAT1 is expressed in astrocytes, there is little evidence about the presence of GAT1 in pyramidal cells, yeah, in excitatory cells. Although some transcripts are at the mRNA level are present there too, yeah. So, and we performed, we recorded inhibitory neurotransmission, yeah, basically spontaneous inhibitory postsynaptic currents. And what you can see here is that the knockout mice actually show increased excitability, yeah, and also the decay time of the spontaneous inhibitory currents is significantly increased, yeah? And this for me is not conclusive, but it's an indication of an astrocytic function, yeah, in the clearance of GABA from the synaptic cleft, yeah? Interestingly enough, when we cross this flux mouse with the VGAT Cree driver line, which actually depletes GAT1 from all the interneurons, yeah, we don't see an effect. Yeah, what the only thing we see is just a decrease in excitability. But at the inhibitory currents level, we don't see a dramatic effect, suggesting that, okay, there could be some compensatory mechanisms that occur in inhibitory neurons, but it's also an indication of the function of this protein that could be not very relevant, at least at 1 month. Yeah, this is has to be said. inhibitory neurons. Yeah. This for us is quite revealing because, of course, we kind of start understanding that potentially some of the phenotypes that we observe in patients could be led by the depletion of Gat1 in EMX lineage cells, yeah, to avoid now citing a specific cellular population. Interestingly enough, and this, I have to say, we can talk about this if you're interested, but when we deplete Gat1 from specifically parvalbumin interneurons, what we get is a decrease. Well, first of all, we get a decrease in excitability, but also we get a decrease in the amplitude of postsynaptic currents, yeah, indicative for me about of a function of this transporter in uptaking GABA to replenish presynaptic vesicles, yeah. So after this, we also became interested in knocking out Gat1 from the full brain, yeah. Also because we are interested in a system that is somehow a bit more dramatic, yeah. And also to know if we can actually correlate what we observe in the full brain compared to different cell types. And therefore, we use the nesting Cree driver line. And here, these are the first experiments we do, so nothing is at the moment confirmed. So, but when you actually get P6 brains from these mice and you perform some stainings to assess the cytoarchitecture of these brains, you can see that basically cortical layering appears to be quite normal, yeah. What we suspect is that there could be some cell densities issues, but this has to be assessed. Yeah. Here we analyzed only a heterozygous mouse. We will go with a knockout mouse, but actually what we can see in the knockout mouse at 1 month is that they show some hind limb clasping, yeah, which is a typical observation of some either motor dysfunction in mice or a stereotype behavior, yeah. And this is for us interesting. The last thing I want to mention and that I'm trying to do is to actually observe the transfer of Gat1 via extracellular vesicles in vivo. Yeah, this is not an easy experiment. Actually, we have to in utero electroporate, yeah, embryos at a very, at around E15, E16, yeah, A14, E15. And actually what we do is to deliver a Gat1 reported plasmid, yeah. So what we are doing is now piloting these experiments. You can see that now we are able to in utero electroporate just a volume marker and see cells that if you're familiar with corticogenesis, you know that at this stage, cells are born, and then when you pick them up 5 days later, they basically migrated to the upper layers of the cortex. Yeah, so this is what we aim to do with the Gat1 reporter plasmid that actually would allow us to see if there is transfer from one cell to the other of Gat1. Yeah. So I will give you just a few conclusions and perspectives because we have many plans, but we have a lot now in the oven. Yeah. So what we know until now is that Gat1 is secreted by neurons in the cortex of mice and human cerebral organoids. We know also that Gat1 mutations affecting protein transport also affect Gat1 presence in EVs, specifically the A288V accumulates in the ER and is never secreted. And depletion of Gat1 affects inhibitory currents in a cell type specific manner. Yeah, this we already know. What we really want to know is to really dissect and in a more detailed manner how this could impact behavior. And indeed, we will perform also behavioral experiments on these mice, but we also want to do what we also want to do is to test the functional roles of the vesicular Gat1. We want to do this teaming up with, initially we thought about Professor Sucic, who could perform GABA uptake assays not on the cells, but on the vesicles. Yeah, we really want to know if the transporter is working on these vesicles as we think it should. We also want to test, yeah, exactly. This is basically also the same, what I was saying, but we want to test in vivo transfer of Gat1 secretion and uptake by neighboring cells. But also we want to in vivo administer EVs, yeah, containing and lacking Gat1 and perform both electrophysiological experiments, but also behavioral analysis, yeah. And I will just conclude saying that this is our group, which is continuously growing. At the center of the picture, you see our PI, Professor Gaia Novarino. And this is me here. And we are a great group that actually we talk about a lot about science. We brainstorm a lot, and it's really nice to be here. And I thank you a lot for your attention. Yeah. And if you have questions, please.
Yeah, there are 3 questions in the chat.
Oh, sorry if I didn't.
No. We can maybe get to them now. Sam, are you there? Would you like to ask your question?
Yes, I'm there. Question was simple. You were trying to resolve. So firstly, very nice presentation. Very informative. Thank you. Question is, have you tried 3D sim approach to better resolve and track both the transporter and the vesicles?
Could you please repeat the question?
Question is, have you tried 3D sim microscopy to resolve these EVs?
Yes. Yes. So initially we thought about this, doing 3D. We had the assumption that this protein is in the membrane of these vesicles, yeah? So initially for us was not really a must. Of course, we could go for it, yeah? So yeah, it's just a matter of, I think, optimizing it. But yeah, we stopped this because we assume, and actually we have evidences by doing immunoprecipitations, for example, that we can precipitate Gat1, yeah, from the vesicles without disrupting the vesicles. So basically you do an immunoprecipitation with that detergent and you get Gat1, yeah, indicating that Gat1 should be at least in the plasma membrane, yeah.
Thank you.
But thank you. I don't know how to read the questions from, maybe I should stop. Now, yes.
There we go. Kai, would you like to ask your question?
Yes. So very quickly, are there any previous examples of membrane proteins traveling from one cell to the other one and maintaining functionality?
Yes. So at least for prion protein, there's a lot.
Yeah, sure. That's of course. Yeah, but I mean for membrane, like channels, transporters, anything like prions are really different. Like
maintaining their function, it's difficult because the fact that I mean you need a knockout to actually address the function specifically to that protein, because these vesicles contain thousands of proteins.
Franco, I'm sorry, I need to leave. Actually, there's another meeting, but I mean, I'm just asking you, is there anything for AMPA receptors, glutamate receptors, you name it, sodium channels, anything? Is anything ever transported from one type of neuron to the other one?
Yes. We recently submitted a preprint now from actually my former lab where we see AMPA receptors and NMDA receptors in extracellular vesicles. The functionality.
And they induce then their own anchoring somehow.
Well, the functionality, we didn't address it to the protein itself. We know that vesicles mediate actually AMPA receptor functions in target cells. We just don't know if it's due to the AMPA receptors in the vesicles, yeah, like specifically.
Exactly. So, I mean, one would think that if you have an exogenous protein, so it just leads to crowding of the native ones. So there are many ways of interpreting. I have actually have conceptual difficulties understanding how a protein which comes from the outside is integrated in a meaningful manner into the brain's machinery. I mean, I think it messes up things completely, but that's just my Like
No. We also have this question, this
already,
issue,
yeah.
right? We need to understand. I mean, you know also that the orientation of Gat1 is also a
Yeah.
problem. Yeah, we know
Absolutely,
that
yeah.
it acts also as a in the reversal mode. So
Yes.
I think the orientation has to be. I mean, we need to work about this, but we also
Yeah.
have the same issues, though.
Yeah, all right. Okay, good luck. I need to leave. So yeah, it was
Thank
great.
you very much.
Thanks a lot. Yeah, bye.
Chris, do you want to ask your question?
Sure. Excellent talk, Franco. I just had a quick question about the nature of the partitioning into the EVs. I'm not an EV expert, but based on the findings that you'd reported, it sounds like there's evidence to suggest that for Gat1 to localize into the EVs, it first needs to be localized to the membrane, which makes a lot of sense. Do you have any evidence to suggest that that's sufficient for it to get into the EVs, or whether there's another domain that's necessary to mediate that? Are there any variants that have come to light that seem to function and go to the membrane but don't end up in the EVs?
No, no. The answer question is no. We don't have evidences of that. What we think is that during the endocytic process, then it makes part, or it goes actually into the multivesicular body system that then fuses back to the plasma membrane. We have now preliminary IPs, yeah, where we did also mass spectrometry analysis, and we see proteins that are involved in this process, yeah, with the wild type version of Gat1. Regarding the mutants, I think we should test some more of them to actually have clearer ideas, yeah, if this is the case. But I think it's a must. Yeah, we should do that. Yeah.
We have two hands raised. Nicholas, would you like to ask?
Professor Trish, hello.
Hi, Franco. So nice to see you. Congratulations on what looks to be like a lot of progress.
Thank you.
I'm sorry I missed the beginning of your presentation. I was curious about the motivation. Like, so I think it's clear that Gat1 is in these extracellular vesicles. But my question is there something special about Gat1 being in those vesicles, or could those vesicles just grab any protein that's on the membrane and sort of, what evidence do we have that there's a specific enrichment of Gat1 on those vesicles versus other membrane proteins?
Yes. So I was expecting to have clear ideas by now about this question. We wondered that, yeah. So the fact that we see this developmental change in the presence of Gat1 in these vesicles suggests actually that is a tightly regulated process, yeah. That is not something stochastic. Actually, the levels of Gat1 increase in development, yeah, but the presence of Gat1 in these vesicles decreases, yeah. So there has to be something special at P0 or at P2 between this time window that makes the presence of Gat1 in vesicles important, yeah. We still don't know. We hope, or I hoped actually, that with the different Cre lines, I could answer this question, yeah. But I know now that interneurons are not the only actors in this secretion, that actually astrocytes are also secreting Gat1. So for me, at the moment, what I would say is that secreted Gat1 could be a signal, yeah, from cells to others to actually say either you have to come here or either, hey, you have to become active, yeah. I don't know. It's just an assumption. But what I truly believe, and this is why we are doing these in vitro experiments, is that the presence of Gat1 in these vesicles could be a tool, yeah. And this is what we want to do. We want to say, okay, nature is offering Gat1 in vesicles. Let's see what happens if we use these vesicles, yeah. Thank you for everything. Your mice are wonderful.
And now Carol has a question.
Hi, Franco. Am I saying your name correctly? Great talk. I'm a postdoc in Dr. Kang's lab, and I wanted to ask you a question about the EFIS data that you have. So the changes in decay that you see in your spontaneous events, that makes sense because I've seen the same thing in the S295L mutant mice. And I also know that Dr. Kavanaugh saw some changes in excitability of neurons. So when you saw that, that also kind of checks out. But I had a question. When you saw changes, specifically, it was an amplitude, I believe, specifically when you delete Gat1 and parvalbumin neurons. Why do you think you see that only in that population, but when you kind of touch a broader population, like with VGAT, which also includes parvalbumin interneurons, why then you don't seem to see those changes? Do you have any thoughts on
Beautiful.
that?
Beautiful. I thought like a week about this. The only answer that I have at the moment is that while VGAT3 actually peaks very early, yeah. So VGAT3 expression, it's even I mean could be also embryonical, yeah. While embryonic, yeah. While P-valve Cree is actually postnatal. So I truly believe that what we are seeing in VGAT is a compensatory mechanism that actually when interneurons are born without GAT1, they can compensate somehow, yeah? While P-valve actually that they are already there and in a way it's not an acute depletion, but it's much more difficult to compensate probably. There you sense the function of GAT1. What we will do is to now to address this question is to go back to P5, for example, and do the same experiment, yeah, to actually see at least with EMX and VGAT if we actually can capture some differences. But yes, we were puzzled too, yeah.
What age were you looking at in your experiments?
At this data is 1 month, yeah.
Okay, that makes sense. Yeah, so I guess about 2 weeks or so, it was that the parvalbumin promoter was on.
Yeah.
That makes sense. Yeah, well, I think actually that makes sense. I think that experiment was pretty sound.
Okay,
Thank you.
Thank you very much. Oh, thanks to you.
We have like 2 more questions, but Chris, do you want to go real quick? And then Kevin, do you still want to ask your question or follow up later?
I can go real quick. Franco, what you'd mentioned about the changes developmentally in the levels of GAT1 and the level in the EVs had just piqued my curiosity. How much is known just about the natural phenomenon of EVs developmentally? And like, I mean, is it possible that there's like some diversity of EVs or like the amount of EVs being secreted by different cell types is also developmentally controlled? So by making a statement about how much is in EVs in total, it could be reflective of there being some change in some underlying heterogeneity of that EV population. How well are you able to stratify? Is it known that there's different EVs? Or do you
Yes.
see what I'm
Yeah.
asking? Yeah.
So, I mean, your question is totally fair. I mean, this EV population is I mean, we have the big population EVs, yeah, that even the biogenesis of these vesicles is different, yeah. I proposed here an endocytic exocytic pathway, yeah, via the multivesicular body, but some of these vesicles are also originating just by pinching off the membrane, yeah, and these are called microvesicles, and they are bigger. Others are apoptotic bodies, yeah, and of course, apoptotic suggests that these are dying cells, yeah. However, the markers that you can have, due to the markers that you use, you can narrow it down to a specific subpopulation that stays as a population. We basically focus on the exosomal population by size, yeah, which still is diverse, yeah. This exosomal population is functional. Now we know it, the field knows it, but it's characterized mainly by the presence of some tetraspanis in the membrane, so some membrane proteins, yeah, which, by the way, are some of the positive markers: CD63, CD9, CD81, yeah. So, and we know now that these vesicles could be triple positive for these tetraspanins, but we also know that there are vesicles that are positive only for one of the tetraspanins. So yes, it's infinite. Yeah. What we really want to achieve is at least to narrow it down to a subpopulation. Yeah. And to understand if this process can be regulated. Yeah. this could be sufficient, I would say, for us. But of course, I think the field is now growing exponentially, and to achieve the resolution that, of course, you are asking, I think some years still are coming. Yeah. Yeah, but it's a fair question. Yeah. Thank you.
Okay, last question, Kevin?
Yeah, quick question. You mentioned that you intend to measure the behavior impacts on the mice, and I'm curious, what is your method of behavioral assessment you intend for the mice? Because I've seen a lot of variation on that.
Yeah. So, well, first of all, here we have maybe there's some background noise. So here we have the capacity to at least to measure some innate behavior, but then move to basically the IntelliCage, for example, yeah, where we actually could monitor these mice for some days. Yeah, where we will also perform also some Morris Water maze, some social interaction tests, and ultrasound vocalizations. Yeah, yeah, exactly. I mean, of course, with specific focus on the social tests. Yeah, but once we ruled out all the innate behaviors, yeah. I mean, we have the suspicious now that the nesting Cree could have this motor dysfunction, yeah, this clasping behavior, yeah, on the hind limbs, yeah. So it could be due either to stereotype behavior, but also to a motor dysfunction. So we want to rule out this, and then we will move to more cognitive and social tests, yeah.
Great, thank you.
Oh, thanks to you. We also have the capacity here to, actually, a colleague of mine is measuring in vivo EEGs, and specifically for Gat1, this could be an interesting aspect to actually test. Yeah.
Okay, thank you so much. That was an amazing presentation. After the meeting, we'll send out the recording, or Amber will. I wish you all to have a great rest of your day.
Thanks for your attention to all of you. Yeah.
All right. Thank you.
Bye.